Donc, on va, Jean-François, enfin, je pense que c'est en anglais plutôt. Hein. Ça te va, Jean-François?
C'est pour vous,
ouais.
ça me va. Très bien.
Oui, je pense que pour que tout le monde puisse suivre. Donc, je vais commencer par une petite introduction déjà. Donc, déjà, je switch en anglais. Donc, thank you all very much for attending today this deep distinguished lecture that will be given by our colleague from Canada, Jean-François Masson. So, first, a very short presentation of Jean-François. After studying chemistry at the University de Sherbrooke, he did a PhD on analytical chemistry, plasmonics, and surface chemistry at Arizona State University, and a postdoc on biosensors at Georgia Tech. He's now professor of chemistry and at the University of Montreal, where he developed new plasmonic materials, instruments, and surface chemistry approaches for the detection of a broad range of molecules, which are then translated to functional sensors for biological, environmental, and industrial applications. His research has yielded more than 125 articles and over 10 patents on instrumental innovation for biosensing. He's also associate editor for SERS Sensors, and he has co-founded a Canadian startup in 2015. Don't hesitate to correct me if I am wrong, Jean-François. So the startup is called Affinity Instruments, and he commercialized surface plasmon resonance instrumentation. Jean-François has also received several awards, and he was named Fellow of the Royal Society of Chemistry. And more recently, he was named in the top 100 most influential analytical scientists from the Analytical Scientist platform. So I won't say more, but I will let you the word now. And thank you, Jean-François, for this lecture that you will give us.
Thank you very much for the introduction. Merci beaucoup pour l'introduction. Juste m'assurer que c'est bien en mode plein écran.
Oui.
Wonderful. So it's a pleasure to discuss our research venture on surface enhanced Raman scattering sensors, where we combine machine learning and nanosciences to detect molecules. And I'll show it mostly in proximity of cells, but also for different applications. So just to give you a brief overview of what my research group is doing. Oops, I'm just going to get the laser pointer. There we go. So we're mostly focused on instrumentation chemistry. So we're designing instruments, and thus we need to combine nanomaterials, surface chemistry, spectroscopy, data processing that I'm going to be speaking about today into applications. And these are two very Canadian applications. So where we've field deployed sensors into the nature, and also we've developed sensors for maple syrup analysis in the past. I'm not going to be able to talk about those two projects today, but as you can tell, these are actually quite beautiful pictures of campus here on the left side, and well, the Canadian wild on the right side. So we use nanoplasmonics. In nanoplasmonics, just very briefly, just make sure that everyone understands what's involved with nanoplasmonics. It is the interaction of light with nanoscale metallic materials. I'm going to just mention metallic nanomaterials, although there are other materials that are plasmonics, but I will just focus on metallic today. In the case of metal nanoparticles, a colloidal solution of metal nanoparticles, light can interact with the electrons of the metal, and that leads to absorption and scattering of the light that will be conducive to detect refractometric-based molecules, and also surface enhanced spectroscopies like Raman spectroscopy. That illumination will lead to resonances in the visible range, which is quite conducive for spectroscopies that I'm going to be speaking about today. It can also be on 2D materials, and I'm not going to have time to speak about 2D materials today, but 2D materials will lead to the excitation of Bragg modes, photoresonances, and localized and propagating surface plasmons. And this is mostly used for refractometric and photovoltaics, electrochemical cells, or other type of cells that are commonly used in, or I would say, physical application of plasmonics. So on the nanoscience side, nanoplasmonics will lead to generation of a lot of data. And machine learning is increasingly being used in that respect. In some cases, and I will actually just do a brief comment. Machine learning in the past, when I was a graduate student, was called chemometrics. It has changed to machine learning due to different algorithms that have been used. But in chemical analysis, it has deep roots in the field of chemometrics. In machine learning for spectroscopy, I just distilled down a very, very basic overview of what are the opportunities and the tasks that can be used. So in the case of supervised algorithms, well, we can provide labels to data. So for example, if we have a spectra of a pure compound, we can label it to that compound. And we can train the algorithm to maximize recognition of the features, which are the spectroscopic bands of that compound, to be able to classify these spectra into different identities. So for example, we could classify spectra associated to glucose, to ATP or ADP, for example, using a supervised algorithm and using training data sets for that purpose. You can also use unsupervised algorithms in spectroscopy to provide information on classes of spectroscopic signatures. So one of the most common scheme is to use that with unlabeled data, so data that we don't know the origins, and trying to classify those into categories, for example, cancer or healthy cells. So in that case, we will be using the clustering ability of machine learning to be able to segregate data into different categories. And more recently, reinforcement learning has started to make a little bit of an impact in nanoplasmonics, where we can train a reinforcement learning algorithm to maximize the output by providing a reward to the algorithm. So for example, we could optimize tasks in nanoplasmonics doing so. For example, controlling a synthesis to reach a very specific plasmon wavelength. So we could have a feedback loop that is trained with reinforcement learning to be able to achieve, for example, a gold nanoparticle that has a plasmon resonance of exactly 525 nanometers, for example. So this provides a bit more of a roadmap of where machine learning could have some impacts in nanoplasmonics. And I'm not going to go into all the details of that. There's too much things. But essentially, the bottom line is that the top part, which the orange path, this is an experimental path. So we manufacture nanomaterials. We get a physical nanostructure. We can observe it with imaging data. And then we can observe the plasmon using, for example, UV-Vis spectroscopy. In that case, the machine learning could bypass all of that and link the plasmon data to the manufacturing conditions into a feedback loop I'm going to show you later on. The blue and the yellow pathways are the modeling pathways, which are heavily extensive use, or that are computationally expensive to perform. And those could be bypassed by machine learning as well. So for example, we could have a feedback between manufacturing conditions and nanostructure model. We could predict a plasmon data from that, or a plasmon model of where the plasmon is located by using machine learning. So all of these are actually in the infancy at the moment. And this is a field where I think a lot of development will occur in the future. And all of that needs to be optimized for the applications that will be later on used for machine learning. So one example is to use machine learning for control synthesis. And this is not something we do in my lab. This is an example I picked from the literature, where we have in manufacturing conditions, we do flow synthesis. Well, they do flow synthesis and then get the structure, measure a UV-Vis spectrum, and then provide that in a feedback loop to change the manufacturing conditions to achieve an exact plasmon data. That can be achieved with mixing reagents using pumps and valves to create droplets of reaction. And each of these droplets contain a set of reagents, reaction time, temperature, for example, and it is measured with, here it's a UV visible spectrum. So the UV spectrum gives an indication of where the plasmon is according to a set of reaction conditions. And that can be used as a feedback loop to tune the reagent stoichiometry to be able to achieve the desired properties, or it can be used to train with a high number of data a modeling algorithm as well. So it has two different purposes. It can be used to generate the large training data set that is required to train a machine learning algorithm, or it can be used as a feedback loop to tune the synthesis in flow chemistry. So this is one area where I see that there could be a lot of impact of machine learning is in that continuous flow synthesis compared to batch synthesis. So when we do particles in my lab, we still use the batch approach where we have a flask where we do a single reaction, then we go to the UV vis and see if the reaction was successful or not. And this is very lengthy and very time consuming. But using flow chemistry, we could speed up significantly that. Another area which is closer to my research interest is to use machine learning for sensing. And in that case, one of the neat application of machine learning is to cluster data. And this has been used for a couple of decades using different type of algorithms. So in the case here that I show from the literature, so one can acquire SERS spectra, surface and Raman scattering spectra, use a deep learning algorithm to classify these into different categories. So use the latent space to see what are the clustering of each of these groups. And in this case, it's for a rapid antimicrobial susceptibility test. So in this case, we have controls that are distinguished from the susceptible bacteria from the resistant ones. So in that case, one could discriminate between a bacterial strain that will be susceptible to the antimicrobial agent, so that will be treated by it, to the ones that are resistant that will not be treated by it. So that provides essentially a decision mechanism where one could decide the course of a treatment against a bacterial infection, for example. So now, everyone has heard of COVID, and we've done a lot of COVID work in the past couple of years in my lab. I'm just going to briefly glance through COVID. I think everyone is a bit tired of COVID, so I'm not going to spend too much time on COVID, but I'm just going to show you two examples where we've used classification of data to provide answers in COVID-related research. So usually for COVID, what we look for is either to detect an active infection through the microRNA, which is the PCR test, or through the proteins and antigens, which are the quick or rapid lateral flow test. In my lab, we've been interested in detecting the antibodies for past infection and also to get a good level of a good idea of the level of immunity one has against possible reinfection later on after vaccination or infection. But the two different aspects are quite important. It's important to see who has had COVID and who has an active infection, but it's important also to see what is the susceptibility of a population to COVID to provide data to public health agencies across the world on what are the next steps to essentially take in case of COVID reinfection or a new wave of COVID. So we've done a clinical trial. So we had a validation cohort that consisted of 60 adults of 18 to 70 plus years of age. We followed them for 24 weeks. We've collected blood samples at week 2, 4, 8, 12, 16, and 24. And we've tested them with classical techniques for antibodies. And we've now applied new techniques. So surface enhanced Raman scattering of metabolites and also surface enhanced Raman scattering of ELISA, and thus to be able to see if we can distinguish between infected from non-infected individuals using SERS, and also to see if we can detect the antibodies with SERS as well. So what is surface enhanced Raman scattering? So this is a plasmonic technique where we irradiate with a laser gold nanostructures in our case. And there are locations where the field will be greatly enhanced. So when a molecule binds on that high field region, it will experience a several order of magnitude enhancement of the Raman signature. So for those of you that don't know much about Raman is a vibrational technique that measures a molecular fingerprint. So a Raman spectra has some peaks, and these peaks will be associated with the vibration of the molecule, which is essentially the construction of the molecule. So if we look at each of these peaks, it will be associated to a group on the molecule, and thus every molecule has a unique signature in Raman because each molecule has a different set of vibrations, and thus will have a different spectra. So what we can do is that we can use Raman to create a unique fingerprint of different type of solutions. And what we have done for COVID is that we've wanted to see what are the metabolite profiles. So we've mixed 10 microliters of human serum that were either COVID positive or COVID negative with a solution of nanoparticles. In this case was silver. And then we've dried that on a aluminum slide, and then we were able to measure the Raman spectra of that. So the Raman spectra were first baseline subtracted using asymmetrically squares, and then we rescaled them and normalized them using extended multiplicative signal correction. And then we remove spectral variation to remove spectral variation that could occur in those cases. So what we see is that there's differences between the negative controls, so the individuals that were never infected with COVID, from the one that were infected with COVID and the one that recovered with COVID, from COVID as well. And especially we see differences in the adenine region, the amino acid region here, and arginine as well, and the amide region as well here. So now we could use a clustering ability of machine learning to be able to see if we can distinguish between all of these different type of samples, namely the negative controls and the one that were infected 4 weeks before the collection of the sample, and the one that were infected 16 weeks before the collection of the samples. So if we use a very simple principal component analysis, so that's the classical chemometrics way of doing clustering from decades ago, and we don't see much separation between the negative controls, the week 4 and week 16. But principal component analysis is agnostic to the origin of the samples, and essentially just try to find the greatest variance in the samples. However, if we use canonical variate analysis and then we feed in the principal components as the input in the x-axis and the biological replicas in the y-axis, so a combination of PC and CVA will try to maximize the variance between the between-class variance while minimizing the within-class variance. And we can see that if we apply this, now we start to cluster the negative controls, the week 4 and the week 16. So we can see that there are some metabolite signatures that is different between the negative control and the one from infected individuals. So that gives us a way that could be useful to see past infection, for example, and recovery to some extent. Can also measure antibodies. I'm not going to go too much details of antibodies. I don't want to distract too much the talk from on that. But the interesting aspect that we could see is that by applying a principal component in this case and clustering the SERS data by age, we see that there's an increasing amount of antibodies for older individuals compared to younger individuals. So the red the dots are for 70 plus years of age, 50 to 69 years clustered together. So the two age groups are not very different. And the younger than 50 years of age actually is lower here at the bottom line. The controls are actually tightly clustered here in this case. We've seen that in other studies and also in our own studies with ELISA that there's a correlation between age and antibody production. And there's also a correlation between susceptibility to reinfection and age. So it seems that older individuals have better antibodies than younger individuals following COVID infection. And now we're monitoring that following vaccination, and we should have this data shortly. So now I'm going to jump into the bulk of the talk, where we're trying to use these type of methods to detect cell secretion events. So one of the blind spots in measurement science is trying to detect molecules that are being secreted by cells. So once molecules are inside of cells, we can collect like high resolution and exquisite fluorescence images of that. But when molecules are being secreted, we lose track of those. We could apply some electrophysiology techniques like patch clamp, where we could see the exchange of molecule between cell and the extracellular environment with high time definition. But this is not a molecular technique. We don't have any idea of what molecules are being involved. We just know that there's an activity of the cell under some circumstances. So what we're trying to accomplish is trying to provide a molecular version of that. And in electrophysiology, the way it works is that it provides a output which is essentially spikes of signal, and that is important to notice because we are trying to provide an alternative technique to electrophysiology. So we're going to try to mimic a little bit the electrophysiology data output. So in electrophysiology, they see spikes of activity, and then when they stimulate, for example, here with the compound, the spike rate increases. So they can compare the spike rate between different conditions, and you see the spike rate will increase when you increase a compound that will stimulate the cells. So those are two important points, because we're going to mimic that in our approach. The problem with electrophysiology is that it's low chemical selectivity. You don't have any chemical information. So we need to provide chemical information that would be used with the same frequency measurement, with the same compatible techniques, such that the workflow in physiology should not be changed. So the way we do that is we go back to surface enhanced Raman scattering. This is the same slide as I presented before. Now I'm going to highlight one thing, is that the molecular fingerprint is due to unique vibrational spectra. That means that if you work in single molecule detection regime, which is possible with surface enhanced Raman scattering, meaning that you can detect one molecule at a time, that molecule has a very unique spectra that you could use machine learning to assign that the spectral signature to that molecule. And now if you do that on a very tiny substrate, you could place that close to cells. And then when you stimulate the cells, for example, here with a red molecule, you could trigger the release of other molecules. Let's say the blue molecule here, that could bind on the tip here of the Raman scattering nanosensor and create a unique signature. So by using a microscope that we've designed, you can actually achieve that in real time and with high frequency of measurement. So the way we fabricate these tips is that we use the very same patch clamp like glass nanofibers that are being used in patch clamp, and then we bind nanoparticles on top of that. We use a chemical process early on in this by using just essentially electrostatic interaction, and we can cluster a lot of nanoparticles on the glass substrate that is submicrometric. So that substrate is within about the radius of 200 nanometers. And these particles are plasmonic, so they have a strong response here in the UV phase, and it can perform surface enhanced Raman scattering. That was a little bit irregular, so we've designed a method to make these substrates more regular. So the way we do it is we use polymer chemistry, a more physical method of absorbing the particles on the surface. Without going into too much detail of the polymer chemistry, so we can bind this polymer here, which is polystyrene polyfluorovinylpyridine, on the glass surface of the tip. And the polyfluorovinylpyridine also has attraction to gold nanoparticles. So what we get is a templated method of binding the particles on the surface of the glass capillary. And we get a well-dispersed but dense array of particles all the way down to the tip that is less than 200 nanometers. That provides a strong Raman scattering, but also a reproducible way of making the sensors. We can bind different type of particles on this. So we can bind spheres, and then we can overgrow these spheres to get better signal. We can create asymmetric particles like raspberries or stars, which is not shown here, on the surface of the glass capillary, providing ways to enhance furthermore the Raman signal. So then we place that sensor at the focal point of our Raman microscope. Then we can put the whole assembly in proximity of neurons in this case. And in proximity will be within 10 microns of neurons, so very, very close. And then we can stimulate the neurons using a peristaltic flow. And then we can create different conditions that will stimulate the release of neurotransmitters. So in the early experiments I'm going to show, we use high potassium to depolarize neurons. And then we can use SERS to see hit rates for different chemicals. And we should see in principle higher hit rate for the conditions that is being stimulated, so the high potassium conditions. So we've adapted a commercial microscope to be able to do that. So this was originally an AFM Raman microscope, and we used the piezo stage here of the AFM Raman microscope to hold our glass fiber at the focal point of the microscope here. And then we can use the white light, the former white light illumination pathway to excite fluorescence, which is exciting the fluorescence from the cells or neurons that we capture with a fluorescence imaging camera. And then we have a Raman laser at 633 nanometers that is diverted onto the tip of the fiber, and it's back scattered into the spectrometer. And then we can acquire images very close to neurons. So here we have a fluorescence image of neurons, and the light green sprout here are the axons, and the axon is exactly where the action is going on in neurochemistry. This is where the neurotransmitters are being released. So we want to place a sensor really close to that. So the sensor is drew here because the sensor is not fluorescent, but in the original image, you can see a shadow where the sensor is. So you can locate the sensor very accurately within microns of neurons to be able to measure neurotransmitters in proximity. So then we turn on the laser, and then we see single molecule Raman scattering spectra. So sometimes we have null spectra, sometimes we have weak spectra, and sometimes we have very, very strongly scattering spectra, as is going to come up in a couple seconds here. So this is a pure solution of dopamine, and each of the spectra for dopamine are different because the dopamine, depending on the orientation on the surface, will present a different profile to the laser, and thus will have a different vibrational spectra depending on its essentially orientation. So in the same way as if we take a picture of someone from different profiles, each of the images are different, but it's still the same person. So when a molecule binds on the surface, depending if it binds upside down, right side up, right ways, left ways, it will present different profiles to the laser beam, meaning that the spectra will be different, such that it creates a rich set of vibrational spectra that is well captured by machine learning. So our initial step is to calibrate for each of the molecules. So here we have a set of different molecules. So we have dopamine, ATP, GABA, serotonin, ADP, and a few other molecules. And each of those will have different spectra or different bars. So you can barcode those, for example. And thus, the same way as you would scan a barcode at a grocery store, you can even scan those bars for different molecules. So that was our original way of processing the data. But before we can do that, we need to pre-process that. So the raw data is first screened to remove cosmic rays. So cosmic rays will cause really sharp and intense spikes in the spectra, which are not related to the vibrational fingerprint of the molecule. Then we do a Savitsky-Golay smoothing. We do an air PLS baseline generation. We create a threshold above that baseline. And if the spectra is intense enough, there will be peaks above that threshold. And then we could assign that as being an active spectrum compared to an inactive spectrum. Because as I shown in the dynamic spectra before, some of the spectra will have a null response and others will have an active spectra. So we need to sort out the null or the inactive from the active spectrum, because the algorithm will try to force feed a label to each of the spectra. So we have only to feed the spectra that have chemical information to be able to provide an accurate response later on. So we've designed a software to be able to pre-process the data this way. So I'm not going into the detail of the software, but it generates the baseline, it corrects the baseline, it generates a threshold, and it determines if there's a number of peaks sufficient to be classified as being an active spectra. For example, here on the top, there's only one peak that is above the threshold. So this is an inactive spectra, while the spectra here has a number of peaks that are above the threshold and is classified as being an active spectra. And only the active spectra are later on processed with the classification algorithm. So here it gives you a bit of an idea of the final product of that. So we have here a raw spectra with the baseline generated here in red. And this is the baseline corrected spectra. And we see the peaks clearly above the baseline afterwards. So we have done data processing using barcoding. So we've created the reference spectra as I've shown. We've give them a barcode, and then we've matched these barcodes from the reference spectra to the active spectra to be able to create a time series of detection events. And in that case, what we would generate is that if there was no match at a given point, we would say it's a 0. And if it was a match for a standard, we give a 1 for that molecule at that time point. So what we created is see, the video is not working, but we created a time series of peaks that detects, for example, dopamine in the time course of an experiment. So I will have data that will show you that later on. But now we've moved to use a convolutional neural network. So the barcoding method was working fairly well, but there was some uncertainty because the barcodes were not fully robust, depending on the orientation of the molecule, such that now we've moved to a convolutional neural network algorithm. So essentially, we can train the algorithm quite easily because we measure a pure solution, for example, of ATP. And we just let the sensor run in the pure solution for minutes to hours. Then we can collect tens of thousands of spectra from the pure solution of ATP. Then we can assign the label to ATP, and then we can feed that into the training of a convolutional neural network that I'm going to show a bit more in the next few slides. Then when we apply that for unknown data, we do the reverse process. We have the spectra, the SERS spectra, that has no label. So then we assign the label in a probabilistic manner using a convolutional neural network. With SERS and pure compounds, it's easy to build large libraries such that the training process is not too complicated in that case. Briefly, I'm not going to go too much over the details of the CNN, but it's a classical, it's nothing very fancy CNN. So we have a convolutional layer applying a 2x1 filter. We have a 2x1 max pooling layer. Repeated that twice. Then we have two densely connected layers. And then we have a softmax that will convert the output of these layers into a probabilistic output for the identity of the compound. To minimize overfitting, we've applied dropout throughout the model. So we've dropped 50% of the neurons at different time points to be able to minimize overfitting, which is a concern in applying machine learning for SERS data. So here we have a confusion matrix for a set of neurotransmitters. So we have dopamine, glutamine, ATP, acetylcholine, GABA, serotonin, ADP, and epinephrine, and glucose. And this is normalized to 1. So we see in the diagonal is the proper assignment, is at least 90% for all of the compounds, except for glucose. And glucose is a bit of a special beast where it does not give a very strong Raman signal, and it gives a very complex Raman signal. So there's a bit of cross-reactivity with glucose. And just to give you a bit of an idea of the training set that we've used. So we've used about 70,000 spectra for the training set, and 30,000 spectra were used for the testing in that. And to validate that we didn't overfit, we've used the same data. We shuffled the label before training, and we did not converge into training this data set if we assigned the wrong labels to each of the data. So randomizing the labels prevented us from training it, thus confirming us that the training data set was quite robust. Then we can apply that to detect actual neurotransmitters in proximity of neurons. We see in the basal condition, we don't see much detection events. When we depolarize, we stimulate the neurons. It generates a lot of signal. And here we use the same representation of data as in electrophysiology. So when dopamine is detected at one time point, we assign it a positive value, and thus we see a peak. And then if we don't see it, we assign a 0, thus a baseline. And we can do that for a different set of neurotransmitters. So we see here the co-detection of dopamine and glutamate. And thus, we see that the two are co-released at the same location. And surprisingly, this is the first time that these two neurotransmitters have been detected simultaneously in real time, because the classical electrophysiology experiments would require two different sets of experiments to be able to detect each of these neurotransmitters. So then we can go a little bit more complex. We can detect neurotransmitters from different regions of the brain. So we can harvest neurons from the mesencephalon, which has mostly dopamine and glutamate release. We can harvest neurons from the cerebral cortex, which has a little bit more glutamate and GABA. And we can harvest also neurons from the striatum, which has mostly GABA. So now we apply the same protocol to that. And we can see that from the mesencephalon, we should see mostly dopamine and glutamate, which is the actual case with our sensor. When we go to neurons that are from the cortex, we should see glutamate and GABA, which we also see here and in the stimulated conditions. And finally, from the striatum, we see mostly GABA, a little bit of dopamine, which is not unexpected as dopamine is ubiquitous in the brain. But we see that the surface-enhanced Raman scattering nanosensor provides us a way to detect different profiles of neurotransmitters in different regions of the brain. So now moving towards more realistic model, we want to move inside brain tissues instead of cultured neurons. So to be able to do that, we can insert the sensor inside brain tissue. And then we can also use, for example, optogenetics to stimulate the release of neurotransmitters. So optogenetics is a optical technique that activates light-sensitive channels that will trigger the release of neurotransmitters in this case. So that is a controllable way to provide a stimulus to the cells inside a brain tissue. One of the challenges of working in brain tissue is this is one of the most scattering tissue, and we use 633 nanometers, which is highly scattering in the brain. So what I show here is that from outside of the brain, we get a very nice Raman signal. But once we go inside the brain from spectra 4 to 6, we lose most of our signal. So we're now working to design a new microscope that will use a different wavelength to be able to go deeper in the brain tissue, but yet we can still measure inside of the brain tissue. And here we have one of the initial data that we've collected inside of a brain tissue. And in about 20 seconds, we will see an optogenetic release of neurotransmitters. So in the basal conditions, we see very, very little signal. You have one of the spectra here. The baseline will go up in a few seconds. And this is when the blue light is being shown on the sample. And then we'll see a burst of a release and then going back to the baseline afterwards. So the baseline should go up about now. The release will go now, and we'll see a large burst of signal. And then we go back to basal level that is co-stimulated by the blue light of the optogenetic signal. So now we're working in combining optogenetics with surface-enhanced Raman scattering to be able to provide a complete picture or more complete picture of the neurochemistry profiles being released by these neurons. As a last part of the talk, I want to go into detection of different type of metabolites, so we can apply these same methods to detect different type of metabolites. And the case test I'm going to show here today is trying to detect differences in metabolites between cancer and healthy cells. So cancer cells, for example, like ELA cells, are suspected to have or should have a higher metabolic rate. And this is why they proliferate faster than healthy cells. So that means the chemical profile close to cancer cells should be different than the one from healthy cells because of their different metabolic rates. So we should be able to detect that using our technique. So we have focused on the glycolysis and this TCA cycle of cells. So glycolysis essentially in brief uses glucose as a fuel and expels lactate as a waste product. And the TCA cycle will use glutamine as the fuel for nitrogen source of the TCA cycle and will release ATP, for example. So we can use our sensor now to provide a spatial distribution of these chemicals. Because a sensor being controlled by a piezoelectric stage, we can move it around in 3 dimensions. So for example, we could start with a sensor being very far from the surface of the cells, and thus we would measure the bulk response. And as we move closer and closer to the cells, we would start to see more and more the metabolism of the cell. And in a very crude manner, we should see a decrease of the fuel from the cell. The fuels are the reagents being used by the cell. And we should see an increase in the waste products of the cell. So essentially, the cell eats some molecules and spits out the waste products. And we should see a gradient by that. So we've calibrated, once again, the SERS sensor now for different type of metabolites. So we have pyruvate, lactate, ATP, urea, and a few more. And each of those have a different Raman signature, thus can be classified by using the same algorithm as I presented before. So by using the same type of CNN as I've shown before, we have at least 90% selectivity for most metabolites. We've used a training set of 1,500 spectra per metabolite and a validation of 500 spectra per metabolite. So we had a few tens of thousands of spectra for the whole data sets. And then we can see that the classification here is at least 90% accurate. As I mentioned before, the machine learning tries to assign every spectra to a label it knows. So we have to calibrate for the CO2 independent medium. So the CO2 medium is the buffer we use for the experiment that has, of course, some molecules that could lead some Raman signals. So we have to calibrate for that to assign the background to the background. Essentially, we create a background category to avoid assigning the background to the known molecules. So now we can measure molecular gradients. So we start far from the cell, so that the shaded areas are from 2 millimeters away from the cell. And as we move closer, we start to see here from 100 micron, the data points will show that we see a decrease of glucose here in the black, closer to cell, and an increase in lactate close to cells. So glucose is a fuel, so that's the energy source of the cell. Lactate is a waste product of that. So we see less fuel close to the cell and more waste products close to the cell. And we see that curvature is bigger with the cancer cell. We see a bigger drop with a cancer cell than we see from the healthy cells. That gives us an indication of that the cancer cell has a higher metabolic rate. We can also measure in parallel, and this was done in a single experiment, the gradients of ATP and ADP. So we see more ATP and ADP close to cells. This is normal because ATP and ADP are produced by cells. And we see also a profile from the nitrogen balance. So we see glutamine, which is the nitrogen source of the cell, decreases significantly close to the cells. And we see a slight increase of urea close to the cells. So we can use that technique to measure chemical gradients close to cells. And the next step for us is to use that to provide a susceptibility mechanism of cells to therapies. So what we want to do is to be able to essentially feed the cells with a therapeutic compound. And if the therapeutic compound is active on the cell, we should see a change in the metabolic profile close to these cells. So that could provide us a test to detect the susceptibility of cells to different types of therapies, for example, cancer therapy. In brief, I'm going to show just a couple different examples where we can detect different types of molecules with this technique. So we can modify the gold surface of our nanoparticles using chemical selective compounds. So in this case here, we have a mercaptoboronic acid to detect peroxide. So peroxide is the oxidative agent in cells, or we can detect also pH. And thus we can have a chemically selective method to be able to provide different type of answers close to cells. In the case of pH, we've shown that we can detect the pH in the extracellular matrix, so outside of the cells, and insert the sensor inside of the cell and detect a pH gradient between the outside and the inside of the cells. We can also do the same thing with mercury. So we can detect mercury in different regions of the cell. So in this case here, we detected mercury in the nucleus and the cytoplasm of cells that were exposed to mercury beforehand. And we see differences in the mercury level between the nucleus and the cytoplasm. So we could even potentially do subcellular detection using these type of measurements. So this is the data for the mercury sensing, and we see here that we see a bigger decrease in the signal, so a higher concentration in the cytoplasm for mercury in these cells. So I'm going to stop here and leave time for question. So in conclusion, well, machine learning has great potential for sensing applications, especially in spectroscopy, because it provides a way to predict, classify, and cluster data, which are typical actions that we want to do with spectroscopy. The potential is not fully tapped yet because we are only starting to apply the more, I would say, robust machine learning algorithms into spectroscopy. So while the early principal component analysis methods or simple vector machine methods have played a tremendous role in spectroscopy, now convolutional neural network or deep neural network are just starting to be used, and reinforcement learning algorithms are, there's only a few examples in the literature. So this is there's a massive untapped potential here to provide better data processing methods for spectroscopy. The dynamic SERS measurements that I've shown in the second half of the talk, we've designed the nanosensors, we've designed a microscope, we've applied machine learning as I shown, and we're now starting to apply those in single cell or single tissue and metabolism pathways and trying to address underlying biochemical questions with that technique. Lastly, I need to thank all the research members of the group, also the funding agencies that are supporting us. Well, lastly, I really need to thank you very much for the invitation, and it's been a pleasure to talk about our research today, and I'm looking forward for discussions afterwards. Thank you very much.
Thank you, Jean-François, for this very interesting, very clear talk. I see that the colleagues agree with that. So now we have time for some questions and discussion. So maybe I'll let, is there any question? Maybe, please.
If nobody is starting, I will start with a question. First of all, you are using, okay, I'm Yves Rosenol, data scientist working with Claire on this Raman spectrum. You are using convolutional network after having transformed your signal by removing baseline, making plenty of correction, meaning pre-processing. And I'm wondering what is really, from your point of view, the interest to use convolutional network instead of any other machine technique. I'm thinking more about random forest or things like this. I have a feeling that using convolutional network, you lose this information connected to which type of feature is discriminated, which is obviously of interest. And then it is related to a second question. In your experiment, you have shown that you are using about 10 molecules in order to train your algorithm of classification. And the problem will be what will happen when you will multiply the number of molecules you would like to discriminate. And especially you have shown that you have to work with 100,000 replicates for discriminating about 10 molecules. And if we think about 20,000 molecules or even more, where does it go?
Let's answer the second question first. There will be a limit. We suspect the limit would be somewhere around 30 to 50 molecules. At some point, you'll start to, well, so Raman has a good discrimination power, but it's not unlimited discrimination power. So, I don't think you could provide a metabolic profile of 20,000 molecules like you get with mass spectrometry, because mass spectrometry is a 2D technique where you can do multiple reactions later on. So the data complexity is much, much greater. So it's not in the same realm. So I think it can address specific questions where you know a set of maybe a few tens of molecules that are being involved in a pathway, and thus provide a targeted scope. Concerning the first question, so we've played with both pre-processing and non-pre-processing, and maybe I wasn't clear, and actually I should correct myself. So the pre-processing is to determine which spectra is active or not. We've tried both to feed the pre-process and the non-pre-processed spectra in the convolutional neural network. We do see a slightly better performance with the raw data, which is in line with what you were mentioning. So there are some features that might be lost in the baseline correction. So what we do most of the time is that we use the pre-processing to essentially screen the active from the inactive spectra. But the difference between the two raw and pre-process is not massive. We're not gaining like orders of magnitude better classification, but we're getting maybe 10 to 20% better classification with the raw data. The use of random forests or other type of prediction technique, those would be good if we're trying to identify what are the variables at the origin of the differences. Yeah. At the moment, we're not interested in that purpose. So we're really in a pure classification task. So we want to make sure at time X, are we seeing dopamine, serotonin, or other molecules? We're not too interested in seeing if it in a more of a origin of signal pattern. The reason also is that we're not doing ensemble measurements. Being close to single molecule regime, we have very high spectral diversity in our training set. And this is one task where the neural networks perform fairly well, is when you have a large diversity of features for the same class of labels. And this is why we've gone into that direction. But I fully agree that in some other Raman applications, random forest, for example, would be great. One of the related application, and I'm just starting to get my mind on that, to determine the features that are being relevant into a data set. There's a couple examples that just came out in the literature that uses essentially the first step. They use the latent space representation by essentially using a self-supervised VAE algorithm, so an autoencoder approach, which is different from random forest, but that achieve somewhat similar result. I'm still not very familiar with that, so I couldn't be much more intelligible on this, but this is something that I think I would look a little bit more into in the near future as well.
Thank you.
Thank you. Jean-François, I have a question, maybe I will ask it now. If I understood well, in your in the beginning of your talk, you show the way you have analyzed some samples from COVID patients. If I understood well, you just mixed some silver nanoparticle solution with the biofluid. I suppose you let it dry, and then you analyze the spectra, and you compare with a database of SERS spectra from metabolic profiles, from different metabolites. And my question is, maybe you can correct me, but if you for this database, does it completely depend on the manipulator experiment, the type of nanoparticle you have selected, or do you think it can be universal, like a database you construct and that can be helpful for anybody? What do you think about this problem?
Yeah, so in this case, unfortunately, we didn't apply a database. So that's something we would like to perform, and we're trying to build that at the moment for other application. So in the COVID patient, you're exactly correct. So we just let it dry, a mixture of serum and particles, but we just use a clustering ability in this case. We just wanted to see if we could distinguish between the classes of control patient and COVID positive patients. So we didn't go into assigning furthermore the molecules. What we've done is that we've used the principal components, for example, of the algorithm to see what are the differences between the different classes, and we saw differences at some Raman shift that were associated with some of the molecules, but that's only based on literature database, not on the embedded database or constructed database. Mm-hmm. One of the things is that when you mix a serum with silver nanoparticles, you essentially only measure amino acids and the purines. These are the molecules that bind very well on the particles, and thus every time you try to classify using serum, you'll end up just measuring the different ratios of amino acid and purines for all different applications. So there's a limitation there.
Mm. Thank you. And just for the patch clamp method, in this case, you precise that you did I saw this number, 70,000 spectra for each neurotransmitters. So in this case, you did a database of yes. Spectra. And why did you do so many spectra? Because maybe it's real time, so you have many spectra very shortly, or?
Yeah, so well, it sounds like a large number, but since we're measuring in dynamic fashion and at 20 millisecond time range, so we measure 50 spectra per second. So if you let it run for an hour, you essentially get tens of thousands of spectra. So it's not that much of a burden to collect this number of spectra. Also, I believe it was that was the total number for all of the metabolites. I think it was about 10,000 per metabolite that we collected. In this case, we need a high number because we're in single molecule regime where the spectra will differ in intensity ratios, or it will differ in peak positions depending on the orientation of the molecule on the surface. That's why we need such a large database in this case. It's just because of the nature of single molecule SERS, that it creates such a variety of spectra that you must account for that richness of information. Otherwise, you'll only partially model that. So essentially, the one way to see it is that it's similar to single molecule mass spectrometry. So if you do mass spectrometry and you explode the molecule, every time you explode it, the molecule will fragment differently. But it's the same way with the Raman. So depending on the exact orientation, you'll get a different Raman spectra.
Okay. Does it mean that? Sorry, Jean-François. Does it mean that when you have said during your talk that you are looking for unique molecule and you have a unique signal, you have a unique family of signal representing this molecule?
Yes.
Okay, and so. Just to understand, what is the rate of acquisition you have for your technique when you are imaging a neuron?
So
my question is, are you able to create an image with Raman signal, for example?
We could. We have not done it because the data cube it would create would be massive. So normally what we do is that we station the probe at one place and we stay there. But nothing prevents us from measuring for a period of time at that point and then just raster around a region. We've just not done it yet, but the principle is there. So we collect spectra every 20 to 100 milliseconds. So essentially, that's about the same scan rate as electrochemistry or as electrophysiology. But that would be a wonderful experiment to have a 3D map. But the amount of data points that we would have to collect would be massive, and neurons don't tend to survive that long on the substrate.
Okay, thank you, Jean-François. Is there any other question?
I have just one question,
yes?
If you want. Did you try to use other wavelengths excitation, such as 532 or 785? Because yes. We know, for example, with 785, we can go deeper into simple. Thank you.
Yes, so we have worked on different projects at 785. The reason why we could not do it for the neuron project is that the piezo stage is on a 633 microscope that no longer has 785. 532 with the bilateral sample, we would be too close for the native fluorescence of the cells. So we should see a very high fluorescence background. But at the moment, at the moment is like today, we're building a new microscope that has a wavelength of 1064 nanometers and 1.3 microns to go deeper in the tissue. So I hope that within the next month or so, the microscope will be fully built and we will have the first data points for Raman at the short wavelength IR range. So yes, we are exploring other wavelengths to go deeper in the tissue. This is ongoing work.
Okay, thank you. One last question or no more question? Okay, so I think we can stop here. Merci beaucoup, Jean-François. Thank you very much for this very interesting talk, and we can stop here. And so maybe with the pleasure to meet you in Paris soon.
Wonderful.
Maybe we, Jean-François, si on discute
oui.
un peu, on peut rester là. Ouais. Ok.
Donc on reste non, on change de réunion.
Ouais. Okay.